Picking up here with part B, we are going to talk about what are called ionotropic receptors, otherwise known as receptor channels. They cannot be understated in terms of their importance, from all sorts of neurological issues as well as the heart and well as endocrine organs and so forth. It's one of the major aspects of cell signaling and communication. So here's our trusty figure from the book again. We have the four major groups of cell surface receptors. We've talked about some of these: receptor tyrosine kinases, G protein coupled receptors, etc. And now we're into the receptor channels, again, also known as ionotropic receptors. With the receptor channels, the ionotropic receptors, they can open and close in response to different types of stimuli. It can be temperature, it can be pressure, it can be the presence or absence of a given molecule. We call those molecules, if the receptor is sensitive to it, a ligand, or if you were in the UK, they'd say ligand. Okay, it doesn't have to be on the outside surface of the cell. We also can see the presence or absence of molecules inside cells that open up these channels. But what happens is when that molecule or molecules are present, the receptor protein changes shape, and then that allows ions to pass through the plasma membrane. So we get a binding event. In this case, it's an extracellular ligand. Changes the shape of the receptor, and now we get change in permeability. And then when the ligand leaves, there's a binding and unbinding kinetics that I'm not really ready to talk about at this time. When that ligand unbinds, then the channel closes again. So we get a ligand that the receptor is sensitive to. The gate opens, allowing, this is sodium or calcium here. It could be any number of charged ions. Chloride, for example, potassium's another one. Those are kind of the big ones. Calcium is in there as well. This is the figure from the book. Here's your signaling molecule, the ligand. Again, they show it on the extracellular compartment. There are ligand-gated ion channels that respond to the presence or absence of molecules inside the cytosol as well. Now that movement of ions can do two things. It can change the voltage across the plasma membrane, or those ions, like it's particularly in the case of calcium, can mediate cellular responses by serving as what we call a second message, a cofactor for other enzymes and proteins inside the cell. And again, when that ligand unbinds, the gate closes again. Moving on from the plasma membrane receptors, we now have a family called intracellular receptors. So rather being embedded in the plasma membrane, these are found either in the cytosol, or we even have a subfamily of these types of receptors that are found in the nucleus called nuclear receptors. So they mediate, by and large, slow genetic responses in cells. What happens is we get a signal molecule, and the signal molecule has to be hydrophobic. I mean, it has to be able to get through that plasma membrane. And if you remember from our plasma membrane discussions, the only things that can get through membranes that don't have some kind of a channel or a carrier have to be hydrophobic. So we have a hydrophobic signaling molecule that then diffuses through the membrane on its own and then binds to either a receptor that's found in the cytosol or maybe as deep as within the nucleus itself. So it's got to be small. I would say small and hydrophobic. I mean, if it's hydrophilic, it's going to have a really tough time. It's going to need a carrier or some kind of assistance. But again, small hydrophobic molecules that will then activate the receptors inside the cell rather than binding them on the cell surface. So Think of lipid type molecules, for instance, steroids, okay, estrogen, testosterone, androstenedione, cortisol. All of those are steroid hormones, and they are very, very hydrophobic because they're very, very closely related to a molecule we know of as cholesterol. Thyroid hormones are also very, very small. They're not quite as hydrophobic as our steroids, but yet they're hydrophobic enough. They have a ring on them that's a carbon ring, so they're hydrophobic enough. Again, will move in across the plasma membrane, bind the receptors, and then mediate primarily slow genetic responses. We call these receptor hormone complexes transcription factors. They're a type of transcription factor because what they do is they, even if they're in the cytosol, once bound, they will migrate into the nucleus and then interact with DNA in order to change which proteins, which genetic material is being transcribed by the cell. So it affects the transcription of DNA into RNA. We can also describe it as turning on or off specific genes. Most of those genes are going to encode a protein of some kind, so we can change the way the cell is behaving by changing the protein suite that it has. Um, I would like to kind of give you an example of this just very briefly. In the last, think of the last 10 years of your life. How much you have changed personally in the last 10 years of your life. Well, since many of you are in your late teens or early 20s, that would mean that you went through adolescence during that time. And adolescence is mediated by a lot of different types of steroid hormones, in particular testosterone and estrogen. So think about how much you have changed in the last 10 years. A big chunk of that is due to the protein, the transcription factor activity of the steroid hormones in adolescence. So let's use estrogen as a model system for this intracellular receptor. Estrogen is, again, it's a steroid hormone. It's actually very, very hydrophobic, so it actually has to move through the blood on a carrier protein. It's so hydrophobic it doesn't want to remain in the blood plasma. It will diffuse out of the bloodstream into the tissues, right across plasma membranes, and will bind estrogen receptors. Now, once those estrogen receptors bind estrogen, they form a dimer, if you will. I'm not too terribly worried about that little factoid. But that dimer then will migrate into the nucleus and bind DNA and affect transcription. In fact, there's an interesting story about estrogen receptors is that when they bind the DNA, they actually bend it. They actually flex the DNA in order to make certain sequences of the DNA more accessible to to the transcription factor machinery. They call it a DNA gender bender, estrogen receptors. And so that DNA gets bent, it opens up the structure of the DNA and allows for changes in protein expression. That includes cell growth and behavior. One little thing you might have noticed about this figure is that we have this listed as a cancer cell. It turns out that estrogen can be a very, very important growth factor for certain types of cancers, like ovarian cancer, for instance, and other cancers that aren't even a part of the reproductive system. So estrogen blockers have become one of the primary types of therapeutics for trying to help change the behavior of a cancer cell. Because again, the estrogen is binding to the receptor, it's coming in here, binding the DNA, and then leading to changes in expression, oftentimes associated with cell growth. So if you block that, you don't have cell growth, you don't have as much of a tumor. And I don't want to leave the guys behind, so we have another picture here, and this time it's testosterone. Again, testosterone is a very, very hydrophobic molecule, requires a carrier protein, diffuses out of the bloodstream, right through the plasma membrane. Here's our receptor protein and the cytosol. The steroid hormone, in this case testosterone, binds that receptor, and we get that hormone receptor complex that can now move to the nucleus. Once in the nucleus, that receptor complex will then engage specific sequences of DNA. It doesn't just bind the DNA randomly. It will target specific sequences in the genetic material, and then bind and change the shape in order to affect transcription. With a reminder that transcription is where you read the DNA and synthesize you use the DNA as a template to synthesize a single-stranded molecule called messenger RNA. That mRNA then gets processed and moved out of the nucleus and is then translated into a protein sequence, which is then folded and modified, as we've talked about before, into a functional protein. So our next concept actually involves that intracellular response to a signal. And I just showed it to you in the form of the estrogen and testosterone receptors. That event, once the signal has reached the cell and binds a receptor, the next steps after that are changing the mode of the receptor. So we have a signal of some kind, and we've shown chemicals up to this point, but it could be voltage or it could be ligands, other sorts of things. What happens once the receptor is bound? And again, I just sort of got ahead of this section in those last few slides, but those set of steps after the receptor is bound and activated are known as transduction. We are changing the language from a first message into the language of the biochemistry of the cell. I mean, that sounds really lofty and all that. What we are doing is we are linking the arrival of the signal with response elements in the cell. It's usually a multi-step process, as you saw in the previous slides. One of the benefits of that multi-step process is that you can amplify a signal. For instance, if you have a receptor enzyme, it's not just going to make one molecule after it binds whatever activates it. It's going to make thousands of those, and those things will diffuse and be transported to other locations, often several locations across the cell. That produces what we would call a unified cellular response that has multiple aspects to it. So multi-step pathways provide opportunities not only for amplification, but also for the synchronization, if you will, and control of multiple responses that give us a holistic response, multiple individual responses that give us a holistic cellular response. I know that sounds a little bit complicated, but we're going to try to dial it back into its individual parts. But you get the receptor binding, and then the steps after that receptor binding are known as transduction. They amplify, they coordinate, and lead to a diverse set of responses in a cell. So what happens in these responses? Well, we have sort of three scenarios that can result, generically speaking. One is that we can change the behavior of enzymes and proteins in the cell. All right, change in cellular function. I sometimes call that a rapid cytosolic response. Okay, you would like to see an example of this? Just look at your right arm, look at the bicep, and then trigger it to flex. You have changed the cellular function, causing those muscle fibers to shorten. It's a change in cellular function. A rapid cytosolic response that can happen in fractions of a second or minutes or so forth. The second possibility is that we get those transcription factors activated. That leads to a change in DNA expression. So we're changing transcriptional patterns inside the cell, leading to differential protein abundance inside the cell. This takes longer. I call this a slow genetic response. In human cells, we're talking about on the order of minimum, I mean bare minimum 18 to 24 hours. Sometimes we're talking about decades and lifetimes. Okay. The real truth, though, is that almost every response involves elements of both. What I'd like to do, though, is help you understand that there may be a predominant response. Back to our bicep analogy here. You can trigger that bicep to flex. And if you keep doing that, what will happen is the bicep will be flexing and relaxing, flexing and relaxing. But also, you're going to be inducing over time, if you kept doing that, if you kept exercising, what would happen is that muscle would get stronger and more ready to flex. So while the flexing of that muscle, the contraction of that bicep muscle is a rapid cytosolic response, at the same time, you are inducing longer term changes in gene expression in order to make the cell more functional to flex and more ready to flex because obviously it's flexing a lot. And so transduction involves these multi-step pathways getting us from the arrival of a signal to a coordinated, quote unquote, holistic response. Most of the signals in between the arrival of the ligand or the arrival of the hormone or the arrival of the change in voltage, we're talking about protein function. The biochemistry of the cell, the language of the cell, if you will, is almost entirely protein biochemistry. So here we have an unstimulated pathway, and all of the pieces of the puzzle are inactive at this point. We get the arrival of, in this case, a signal that's not going to get across the plasma membrane. It's going to bind a receptor. This might be a receptor enzyme or a G protein coupled receptor. What happens is when that ligand binds, it causes a change in the shape, the change in the conformation of that receptor, which then allows it to perform some other type of function. And as we described in the cell surface receptors, or even in the intracellular receptors, when we turn the intracellular receptor on, it migrates to the nucleus to serve as a transcription factor. So now this receptor is on. It will now interact with other proteins. Maybe we have a protein that's localized to the inner surface of the plasma membrane. That's a target. And now this gets changed by the receptor and so forth. Now that one changes another target. That one in turn changes another target. But this isn't a one-to-one thing. This receptor isn't just going to affect this one copy of this one protein. It may activate dozens or even hundreds of versions of that protein. And this in turn will activate hundreds of copies of this. So if you had several hundred times several hundred, now we're into the tens of thousands. And if this one activates hundreds of these, now we're into the hundreds of thousands or millions of molecules. That's that amplification step. And also diversifying the response as well. Okay, so it's this stepwise pathway involving proteins. And like you said, like falling dominoes, but it's not one domino to one domino to one domino. It might activate 10 or 100. One of these would activate tens or hundreds or thousands of these, etc., etc. So there is an amplification and what we call a divergence as well. That would say we're going to affect many different cellular processes. Okay. We are transducing the signal from its original first message into the language of protein biochemistry inside the cell. There are several types of transduction mechanisms. We can modify proteins with cleavage or with cofactors, molecules that are created by enzymes and so forth. But one of the most important, well-studied and well-understood mechanisms is known as phosphorylation and dephosphorylation. What happens is we have these phosphate groups. We talk about phosphates on ATP, for instance. These phosphate groups they're bulky and they also have charge. And so they serve as very, very powerful ways of changing protein shape. All right. So we can phosphorylate proteins, change their behavior, and that can lead to a form of signaling. We call them kinase cascades, and that's what the word kinase means. A protein kinase will take a phosphate from ATP and transfer it onto the protein target, and that's called phosphorylation. And kinases are the family of proteins that have a target. So each kinase has a set or very unique specific range of targets. There's an amino acid in that target somewhere that has an OH group on the side chain. And what will happen is the kinase will take that third phosphate from ATP and transfer it onto that OH group. We end up with a phosphorylated protein and ADP. Now the counterpart to kinases are phosphatases, not phosphorylases, phosphatases. There's an enzyme called phosphorylase out there, and it gets really confusing, I understand. Phosphatases are the counterpart. They will have a protein target where that's phosphorylated, and they will pop that inorganic phosphate right off of there. They won't transfer it usually back to ATP. Creatine kinase is kind of a weird example of something like that. But in this case, what we're doing is we're just popping that phosphate off, and we're back to an unphosphorylated protein. Again, these two forms of the protein may have very, very different behaviors or shapes. So protein kinases mediate the adding of a phosphate to an OH group on a protein, and protein phosphatases mediate the removal of that phosphate. And this basically becomes a sort of on-off switch or a molecular flip, if you will, between phosphorylated and unphosphorylated. And this can happen quite rapidly. We can cycle through these in less than a second. Now let's put these kinases into the larger context of signal transduction. So here's up here at the top, here is a ligand-gated receptor of some kind, not a channel. Could be a channel. It could be. All right, let's say it's a receptor enzyme or something. Okay. In any case, it's activated. Now it's going to activate some other relay molecule, or maybe it'll do the job itself. And here we have an inactive kinase. And all of a sudden, with that activated relay molecule, now that kinase is turned on, and now it is free to phosphorylate targets. And so its target is another kinase, and so it phosphorylates that. Now this kinase is active. Well, it turns out this is also a kinase, which targets this inactive form of kinase, etc., etc. Remember how I said about amplification? Kinase isn't just going to phosphorylate one target, it's going to phosphorylate dozens, hundreds, or thousands of these. So it's a very, very lovely way of amplifying through steps, or diversifying, let's say divergence. Maybe we target a couple of different kinds of this kinase that do go to different parts of the cell and do have different targets. Okay, this collectively is known as a phosphorylation cascade, or sometimes they will call it a kinase cascade. Okay, so we turn on the first kinase, and that leads to this sort of amplification, diversification, and finally we'll get to some kind of effector protein. And that effector protein will then in turn be phosphorylated and lead to some sort of specific type of morphologic or enzymatic type response. An example of what we could use for this, I'm just kind of thinking of a couple of different things. Smooth muscle, for instance. Smooth muscle, the myosin, the motor protein in smooth muscle has to be phosphorylated in order for that smooth muscle to contract. Another example would be growth factors, things that cause the cell to grow and divide. This protein here might be phosphorylated and is a transcription factor that then moves to the nucleus and changes the transcriptional pattern of the cell. So we have this phosphorylation, another name for it is a kinase cascade. It's you have to remember though that these kinases don't act in a vacuum. They also have corresponding phosphatases that will downregulate this pathway. As you know, so you're not just turning it on and leaving it on, you're turning it on and then transiently, and then the signal will be turned off by phosphatases. Another kind of common transductional mechanism in cells are known as second messengers. Now, I could say phosphorylated proteins in the previous mechanism are second messengers. That's true. More often, we use the phrase second messengers for small molecules that are created by receptor enzymes and ions. So if the signal arriving at the cell is a first message or a first messenger, like a ligand for a receptor on the cell surface, then the molecules that are diffusing, that are created, released, diffusing throughout the cell, mediating protein function, are known as second messengers. A lot of times they're pretty small. They're very seldomly proteins. I would argue that the phosphorylated kinases in the previous slides are second messengers. All right, it's a different form of the message. It's the second message. Okay, tend to be water soluble because it's an aqueous environment inside the cytoplasm, so ions can work very well through this. Spread by what we believe is diffusion. Second messengers, things like the GPCR activated pathways, receptor tyrosine kinase pathways, again, are very, very common. So we may have phosphorylation cascades, and we also may have second messenger release. Two of the most common second messengers are known as cyclic AMP, abbreviated with a lowercase c, capital AMP, adenosine monophosphate. It's an ATP missing two of its phosphates. All right, that's also been modified to form a ring structure. So cyclic AMP and then calcium, two of the most common second messengers found inside the cytosol of cells that have been stimulated. And so here's calcium. Here is cyclic AMP on the right. Here is AMP, adenosine monophosphate, on the left. The adenosine base, the ribose sugar, and a single phosphate. You can see what happens is that phosphate gets attached to this OH group right here and forms this little ring structure apart from the rings that are already there. Cyclic AMP. They serve as allosteric regulators, okay, allosteric regulators of protein function in cells. Here's an example that we could all be somewhat familiar with if you've ever studied muscle contraction. There are a complex of proteins on the surface of the fibers that contract. This is what's called an actin fiber, in dark purple here, bluish purple. Those are monomers of actin, those individual circles. But wrapped around this actin is a protein complex known as troponin tropomyosin. It's several proteins. About the best way I could describe it is they act a little bit like a garage door. Okay. When that second message arrives, that calcium in this case message arrives, it will bind troponin, which will change troponin's shape, will pull on tropomyosin and pull tropomyosin away from these little black shaded dots on the actin. These black shaded areas, which are covered up over here and not covered up over here, are where the motor protein myosin can bind and pull on those actin filaments. So it's like a garage door. We get the signal, it's received by the antenna of the garage door motor and activates the motor and pulls the door away so that the car can come in or go out of the garage. Okay, that's a kind of a crude analogy, but what we're doing is we're changing the protein shape, which changes its behavior in relationship to whatever it's dealing with, in this case, actin. And so now we'll get to the final piece of the puzzle, the output, if you will, the response activities that we observe in cells in response to A, the signal arriving and binding a receptor, and B, the transduction events. Now we're into the doing and the responding side of the equation. And we talked about this a little bit earlier, the fast cytosolic responses and the slow genetic responses. And so I'm just going to recapitulate some of this. We've seen this before, so I'll move quickly through this. If we have an intracellular receptor, that generally means that it's going to lead to a slow genetic response. The closer to the nucleus you are, the more likely it's going to be a slow genetic response. That's the predominant response in the cells. If it's nuclear, you can guarantee that it's going to be a slow genetic response. So we're going to get some change in cellular activity. It may be a fast cytosolic response, like for instance muscle contraction, or some other relatively rapid activity. All right. So a rapid cytosolic response, cytoplasmic response, or if it's an intracellular receptor, that means it's probably going to lead to a slow genetic response. So slow genetic responses lead to synthesis of new proteins, new enzymes, changing the genetic expression of the cell. Every cell in our body generally has almost all of our genetic material, so we don't want to express all of it at once. What we're going to do is turn on a subset of those in order to address whatever function that cell or tissue has. So intracellular receptors often lead to slow genetic responses, transcription factor responses. Fast cytosolic responses can do this too. I want to show you that these paths do converge, but just make sure that you understand that there are two different outcomes. There's a fast cytosolic response and a slow genetic response. So here we have a ligand-gated receptor on the plasma membrane. Your growth factor ligand has bound. It's activated the receptor. That receptor has then interacted with something that then, in this case, it turns out to be a phosphorylation cascade. We have these kinases that will amplify and diversify the signal. One of those responses may be to phosphorylate a transcription factor. You can see the transcription factor is phosphorylated here. If it's not phosphorylated, it's inactive. And so now that we have a phosphorylated transcription factor, it binds the DNA, changes genetic expression patterns, and we get RNA transcription pattern changes. It isn't always just, okay, we're going to start transcribing mRNA here. That's possible. That's one of the possibilities. Maybe we are shutting down a form of transcription, or maybe we are making transcription more robust. We're making more of something we're already making. We're just making boatloads of it. That modulation is important. It's not a binary system that all of a sudden it's on or off entirely. But again, put the pieces of the puzzle together. If we tried to start from 30,000 feet with a figure like this, it can be very, very complicated. So the way we deal with it is to break down the individual pieces like we have in this lecture, and then work sequentially, like a set of dominoes in a domino cascade. And just to show you how diverse and amazing this process leads to, the emergent properties of what may feel like a relatively simple system. These, by the way, are all the different possible types of disorders that occur with things like estrogen. Okay. So lots of divergent, various types of cellular behaviors and responses, all to one molecule. It's pretty amazing stuff. It's pretty amazing stuff. But as complex as it might seem, if you work on the individual pieces and then start working on how to connect those pieces and where those pieces ultimately lead to slow genetic responses or fast cytosolic responses, the elegance of the system becomes quite apparent. And lastly, I would just like to say that if this lecture doesn't help you in the way that you would like to be helped, there are lots of resources out there, wonderful resources. Khan Academy is no exception to this. If you're looking for additional definition or material, I encourage you to get out there and engage in it. So thank you for your time, and this is the end of Chapter 11, and we'll see you again soon. Goodbye.